Hey everyone, welcome to TutorZone. Today we are going to talk about red cell inclusions. But before we dive into the topic, let's talk about red blood cells. Red blood cells, in short form RBC or erythrocytes, are the main cells in blood. They are kind of like pink or like light purple color, and their shape is biconcave shape. So you can see the paler region at the middle of the cell surrounded by a dark colored area. We are normally comparing the size of a normal red blood cell with the nucleus of a small lymphocyte. So the size of the nucleus of a small lymphocyte is equal to the size of a normal red blood cell. Then, what are red blood cell inclusions? Red blood cell inclusions are the extra things found inside the red cells, like abnormal structures or particles. Okay, and they are not doing a specific function, like the organelles in a normal cell. Having these structures inside red blood cells can help doctors figure out specific diagnosis. So, spotting these cell inclusions in a blood smear is important. Let's see what are the types of red cell inclusions. First one, basophilic stipplings, Cabot rings, Howell jolly bodies, Pappenheimer bodies, malarial parasites, nucleated RBCs, and the last one is Haynes bodies. Basophilic stipplings, as the name suggests, refers to the blue colored dots seen inside the red blood cells. These dots evenly spread throughout the cytoplasm, and these are made up of what? They are remnants of RNA precipitates. So they are evenly spread over the cytoplasm of the cell. So we can see these kind of cells in lead poisoning and in hemolytic anemias, and also in the conditions where pyrimidine 5 nucleotide deficiency. So this is the enzyme which is responsible for the degradation of RNA. If this enzyme is deficient, then it will cause the RNA to precipitate and appear in the cytoplasm of the red blood cells. Cabot rings, they are very fine single or double thread-like loops seen inside the cytoplasm of the red blood cells. So they can appear either ring-shaped or in figure 8 shaped. You can see the both ways in these two pictures. The first one, this is the ring shaped one, and the second one, it's the figure 8 shape. They are mitotic spindle remnants, and we can see these kind of structures in megaloblastic anemia and as well as in thalassemia. Howell jolly bodies are small, dense, round, nuclear remnants usually observed at the periphery of the red cells. So these are nuclear remnants, that means they are consist of DNA materials. And these structures can be seen in conditions such as thalassemia, megaloblastic anemia, hemolytic anemia, and in patients who have undergone splenectomy. Next one is Pappenheimer bodies. These are small, irregular, blue-colored granules which are clustered near the cell periphery. So these are in cluster form, and they are also blue in color, and they don't have a regular margin, they have a irregular appearance, irregular margins. So these are iron granules, and they are observed in conditions like sideroblastic anemia, hemochromatosis, hemoglobinopathies, and in thalassemia. In malarial infections, we can see two stages commonly inside the red blood cells. Normally in blood smears, These two stages are the most common stages. First one is the ring stage, you can see in the first figure, and the other one is the amoeboid stage. These stages represent the life cycle of malarial parasite inside our red blood cells. Here we can see the nucleated red blood cells. These are the immature form of red blood cells with the nucleus. When the bone marrow is working hard to make more red blood cells in moderate or severe anemias, these immature cells can sneak into the bloodstream. So these cells are known as nucleated red blood cells, which have a size similar to the normal red blood cell but with an acentric nucleus. Last one is Hen's bodies, but we can't observe these structures in a normal blood smear stained with Leishman stain. We need to use a supravital stain like methylene blue to observe Hen's bodies. These are precipitated denatured hemoglobin during oxidative injury. Therefore, these cells can be seen in conditions such as G6PD deficiency and in unstable hemoglobinopathies. These Hen's bodies reduce the flexibility and shape of the red blood cells, making them more likely to be destroyed by the spleen. When splenic macrophages clean up the damaged parts of the red cell membrane, it leads to the creation of white cells. So in this figure, you can see white cells. So these cells can be found in peripheral blood smears stained with Leishman stain as well. Now we are going to summarize the things that we have learned in this video. We have covered different types of red cell inclusions, their composition, identification methods, that means the appearance in a blood smear, and the associated conditions. The first one is basophilic stipplings. They contain, these are blue dot-like structures which are evenly spread over the cytoplasm. They consist of RNA, can be seen in lead poisoning, hemolytic anemias, and pyrimidine 5 nucleotidase deficiency. Second one, cabot rings. They are parts of mitotic spindle. You can see them in ring shape or in figure 8 shape. Can be seen in megaloblastic anemias and in thalassemia. The next one is Howell-Jolly bodies. They consist of DNA, can be seen in thalassemia, megaloblastic anemia, hemolytic anemias, and in post-splenectomy. Then Pappenheimer bodies, they consist of iron granules. So they are irregular in shape and you can see in cluster forms at the periphery of the cells. Can be seen in sideroblastic anemia, hemochromatosis, hemoglobinopathies, and in thalassemia. And the last one, Hen's bodies, they consist of denatured hemoglobin, can be seen in G6PD deficiency and in unstable hemoglobins. That wraps up today's video. Thank you for watching. If you found this information helpful, don't forget to subscribe and share with your friends. Stay tuned for more valuable content. Until next time, take care.